So as previously promised, brought here at great expense from SRTC, I unchained him from the bench. George Kaysen, graduate student in my lab, will be talking about single-stranded DNA viruses. And in theory, it's all being recorded, and in theory, it will all be posted to YouTube. We'll see if that happens. So, George, all yours.
All right, I have to get used to the glow of the screen in the dark here, so this may take me a few minutes to get comfortable. But I'm George Kaysen. I'm one of Dr. Stedman's students, and today we're going to talk about small single-stranded DNA viruses. So we're going to go through kind of the common things that you guys go through in a virology lecture. So we're going to start out talking about parvoviruses, and we're going to talk about kind of the standard things that you guys talk about, where they come from, what they look like, how they infect cells, what their genomes look like, how they make mRNAs, proteins, and how they replicate their genomes. We'll look at the differences between dependoviruses and autonomous parvoviruses. We'll just look at that briefly. We'll look briefly at some applications. And then we will talk about some of the stuff that we work on in our lab. And there's your first typo of the day. And again, for the stuff we work on in our lab, we'll look at genome structure, potential structures, how these viruses replicate, and the origins. All right, so real briefly, hopefully this is review. I just wanted to put this up to remind everybody of cell cycle. Probably learned about this in principles of biology and then maybe in a little more detail in molecular biology. Just as a reminder of what goes on during the cell cycle. So a typical cell has G1. This is where metabolic things happen, where a skin cell performs skin cell functions. You have the S phase. This is where DNA replication takes place. So all the enzymes that are needed for DNA replication are, in theory, localized to the nucleus during this phase. You have G2, the cell gets ready to divide, and then during mitosis, cell actually divides. And all these referred to as interphase. No, we're not talking about cell biology per se today, but the viruses that we talk about today are going to be dependent on having cells in this S phase for their replication. So they are dependent on the enzymes that are present during S phase, specifically the DNA replication enzymes that are present in cells during that time. All right, so the origins of these small single-stranded DNA viruses. We're going to start out by looking at small single-stranded DNA viruses that are relevant to human or mammal life, and we'll look at some that infect plants. Then at the end, we'll look at some that we don't know what they infect. So the origins of these particular viruses, lab rodents were injected with tumor material, and then screens for viruses were performed, and it was assumed that viruses that were isolated from these tumor-injected mice, that these viruses should be oncogenic, right, giving rise to cancer. It turns out that the parvoviruses actually aren't oncogenic. They're simply oncotrophic, which means that they have a tropism for rapidly dividing cells, which cancer cells, right, are out of control dividers. So most of these parvoviruses were discovered more or less accidentally. So again, you inject tumor material, you find parvoviruses. If you look for when adenoviruses were discovered, these adeno-associated viruses were also discovered as a contaminant of those preps. And so a lot of these viruses were discovered just more or less by accident or as a byproduct of discovering what people might view as more interesting or human relevant viruses, because until very recently, these dependoviruses weren't known to cause any significant health effects in humans. And we'll talk about what specifically a dependovirus is here in a second. So again, the replication of these viruses is dependent on being in a specific phase of the cell cycle. Again, that specific phase is the S phase, because you have all the necessary enzymes needed to duplicate or replicate DNA during that phase. So autonomous versus dependoviruses. So the autonomous viruses, the autonomous parvoviruses, don't require infection with another virus. So they can infect a cell, and they can hang out and wait until that cell enters S phase, and then they can duplicate their genomes. These dependoviruses, as the name suggests, they are dependent on infection with another virus to push that cell into S phase. So the adeno-associated virus is dependent on adenovirus, and there's a model of the AAV up front here, one of Dr. Stedman's 3D printed models that I'm sure he's shown you others of. If you want to play with that after class, you can come up and look at it. I think it's easy to see, or easier to see your axes of symmetry when you can actually hold something and move it. So probably the most common parvovirus that people are familiar with, if you've ever had a puppy, your puppy was hopefully vaccinated and hopefully didn't get canine parvovirus. So it's a disease that causes diarrhea, intestinal distress in young dogs, and it's generally, I think it's something like 75% fatal in young puppies when they get it. So we'll generally focus on minute virus of mice because it's the most well-described autonomous parvovirus. When we're talking about dependoviruses, we'll be looking at adeno-associated viruses. So if you guys have questions, just get my attention somehow, and I'll do my best to answer those questions. So standard stuff, structure of these viruses, T equals 1 naked icosahedral capsid from 60 subunits. Because you guys have had a few of Dr. Stedman's tests, you know that he likes questions that go beyond that. And so from this information, you can probably, hopefully arrive at the H and K values. You can imagine, or rather you can compare this structure to other viruses that you've talked about, because you know that he also likes comparison questions. These viruses are small, so 28 nanometers in diameter. Parvus is Latin for small. So this 5K linear, and we'll see why linear is in quotes here in a minute, 5KB linear single-stranded DNA genome allows these viruses to maintain a very small physical structure. So down below here you have cryo-EM of our model up front here, and you'll see that in some places you have kind of a dark turquoise color. And so that dark color on the cryo-EM structure suggests, or strongly suggests, that the DNA is actually, the genome associates with the capsid proteins when that DNA is packaged inside the virion. All right, so you guys go through this for most viruses, I think. Attachment and entry for minute virus of mice binds to sialic acid. Again, common questions, what other viruses bind to sialic acid? Go back so you can make those comparisons. The adeno-associated virus, it's got a whole bunch of different receptors. Once these viruses get inside cells via endocytosis, they then have to move to the nucleus, where they get inside the nucleus via nuclear pores. Remember that they are dependent on cells being in the S phase, where S phase enzymes, they're in the nucleus. And so these viruses can get inside lots of different cells. So canine, and we'll go through this on the next slide actually, different viruses can get inside cells, but in order to truly infect a cell, these viruses have to get their genome into the nucleus. And so uncoding at the nucleus is the critical step, as opposed to actually getting inside the cell kind of at the beginning. So that brings us to permissive versus non-permissive cells. So cells that contain the correct extracellular receptors that allow a virus to bind and enter that cell, we refer to as being susceptible. So if you back up the slide here, for minute virus of mice, the correct extracellular receptor is sialic acid. So a cell that has sialic acid on its surface, in theory, could allow MVM inside, so we would say that that cell is susceptible to MVM. Cells that have the correct machinery for replication are called permissive. And so for a successful natural infection, the cell needs to be both susceptible and permissive. So the virus has to be able to get in, and once that virus is inside, the cell either needs to have the correct machinery or it needs to be able to be reprogrammed to have the correct machinery to duplicate that viral genome, make viral proteins, and send that virus out to find a new host, potentially at some point. And so again, this uncoating at the nucleus for the parvoviruses, for the small single-stranded DNA viruses, is the critical step. So canine parvovirus, which we talked about just a minute ago for dogs, can get into cat cells, but once it's inside those cat cells, it won't actually replicate. So we would say that cat cells, feline cells, are susceptible to canine parvovirus, but they're non-permissive because they don't allow replication. But if you just take naked DNA, naked viral DNA, and transform that into feline cells, take naked canine parvovirus DNA and transform that into feline cells, that DNA can, in fact, replicate, which further implicates the uncoating at the nucleus as being the critical step to tropism here. So canine parvovirus can enter permissive and non-permissive cells. So you can get inside. If you can get inside a cell as a virus, we would say that that cell is permissive. If you can replicate, that cell is So canine parvovirus can get inside permissive cells where you can replicate your genome. You can also get inside non-permissive cells where the genome replication is not possible. All right, so how are these small single-stranded DNA genomes organized? This is probably pretty similar to a lot of the maps you guys have looked at for other viruses. They're small, relatively small genomes, about 5 kb, with relatively few genes, which results in lots of splicing in order to make different gene products. And then you have a few examples of alternative start codons. So this box here on the lower right, for the structural proteins, you can see 2 different proteins being made from the same mRNA. So you have one start codon here and a second start codon at the right side of the little box. So generally, the non-structural proteins, the proteins that don't make up the virus, don't make up the coat, are located on the 5' or left half of the genome. And then your capsid or structural proteins are located on the 3' or right half of the genome. So in the case of minute virus of mice, you have 2 promoters that drive mRNA expression. In the case of adeno-associated virus, they have an additional promoter for a total of 3 that drives expression. So you can see the mRNAs here in the long lines. The proteins that are made from the mRNAs are in boxes. The gray boxes are non-structural or replication proteins. Your brownish boxes are structural capsid proteins. These V's, inverted V's, I guess, represent introns. So you have a lot of information contained in one picture here. All right, so I mentioned that, or I had linear in quotes at the beginning, right? So these genomes are technically linear, but they have these regions of self-complementarity at the 3' and over here at the 5' end of the genome. And so they fold back on each other, and you'll see that your correct Watson-Crick base pairs are established here, CG, AT. And so this is referred to as a terminal hairpin. And so these genomes are linear, but they have extensive secondary structures, especially at the ends of the genome. And these secondary structures accomplish a couple things. They enable self-priming, which we'll talk about in a minute. So you guys have talked about different DNA replication strategies, or genome, I guess would probably be a better phrase, genome replication strategies. So you've probably looked at protein-primed DNA primed. Now we're going to look at self-primed genome replication. They also, having these terminal hairpins also overcomes the problem of DNA end replication. We'll look at how DNA end replication is a problem in a couple slides. Okay, so how do we get from this single-stranded genomic form of DNA to a form of DNA that can actually make mRNA? That mRNA can in turn make proteins. Well, again, because you have these terminal hairpins, the self-complementarity, at one end you have a free 3' OH. From molecular biology, if you took molecular last quarter with Dr. Sedman, a free 3' OH is exactly what DNA polymerase needs to extend from, right? So here's our free 3' OH. Now a cellular DNA polymerase can extend the 3' end out through this terminal hairpin. You'll see that it unwinds that hairpin. And if you follow along, the A, B, A', and C are simply sequences. Those letters are there just to help you keep track of where stuff ends up, and those will become important for replication here in a minute. So cellular DNA polymerase extends from the self-primed 3' OH. It unravels this terminal hairpin. Now you have double-stranded DNA that can serve as a template for transcription. So you can make your appropriate mRNAs from this template. All right, so we've seen how single-stranded DNA gets converted to double-stranded DNA, right, in the nucleus by cellular DNA polymerase. Now, so it's both cellular transcription factors and viral transcription factors can bind to the promoters. And so you have, in red boxes, you have examples of cellular transcription factors that will bind to these viral promoters. And in green, you have viral transcription factors. Yeah?
So is genetic information coded on both regions? Once you elongate, right? Or once the polymerase attaches itself, is genetic information right on both sides?
The question was, once you end up with this double-stranded form of DNA, do both strands have open reading frames? Is that the correct question?
Yeah, just like information. Sometimes some viruses have like genes that are coded on one strand or not.
Yeah, so the question was, and tell me if I'm right, is there information coded on this strand and on this strand?
Yeah.
I would imagine not. I don't know for sure. Only because the way that these are represented would make me think that the relatively few mRNAs that are produced probably come from the same strand. I don't know for sure, though. Okay, so you have both cellular transcription factors that bind promoters in red, common ones E2F, SP1, TBP, and here's your transcription start site. You also have viral transcription factors in green that bind to promoters. I mentioned that for VP2, which is the major capsid protein for these viruses, and VP3, which is a minor capsid protein, you have alternative start codons. So they come from the same mRNA, same protein sequence, except VP2 has this additional, slightly darker box portion of mRNA that results in protein here. Wrong button. Okay, so we've seen how single-stranded DNA gets converted to double-stranded DNA, how that double-stranded DNA then serves as a template for production of mRNAs. Now we're going to look at how these genomes are replicated. So I mentioned that DNA end replication is a problem. So replicating linear ends of DNA is a problem. So there's a couple ways that organisms have overcome this problem. The simplest maybe not simplest, but most obvious, is that they get rid of DNA ends by having a circular chromosome. Other options are using enzymes like telomerase and reverse transcriptase. So just briefly, how is DNA usually replicated? You usually have an RNA primer, which leaves a free 3' OH. You can extend from that 3' OH, incorporate your correct complementary nucleotides until you run into your RNA primer or until you run to the end. Those RNA primers get removed, and now you have gaps. At this gap, this internal gap, you still have a free 3' OH from this DNA here that you can extend from, but at the end, there's no free 3' OH to extend from. So again, you can get rid of ends by circularizing, or you can use specialized enzymes to fill in these gaps here at the end. Or in the case of these small single-stranded DNA viruses, you can use hairpins. Okay, so how do these genomes get replicated? So this big gray circle, so if you guys have the textbook, I don't think that this big gray circle is in your textbook. I think I went through and added this. So if you look in your textbook and don't see this, don't worry. And so what this big gray circle is the rep protein, the replication protein, the protein that mediates replication in adeno-associated virus and our other single-stranded DNA viruses. And we'll look at what this protein does and doesn't do here in a minute. So here's our single-stranded genomic DNA with terminal hairpins. Here's our free 3' OH. Here's the 5' end. Here's the rep protein associated with the 5' portion of this genome. Cellular DNA polymerase comes along, finds our free 3' OH, and extends out through the end here. You can see that it breaks down this hairpin structure here. So now we have a double-stranded form of DNA. That double-stranded form of DNA can, like we showed on the last slide, can serve as a template for making your mRNAs, or it can be duplicated. So once you have this double-stranded form here, the rep protein can move to the site exactly opposite where this original free 3' OH was and introduce a single-stranded nick. So it doesn't completely cut through both strands of DNA. It only cuts here. This rep protein can then unwind this terminal hairpin, so it nicks and unwinds through helicase activity. Now again, you have a free 3' OH that DNA polymerase can extend from. You can see now that you've replicated your DNA ends without having the need for things like telomerase or being circularized. So once this happens, your hairpins will reform and the cycle can continue. If you follow along with your A and A prime, C and C prime, what you'll notice is that starting from here to your progeny, so moving from your parental DNA to your progeny DNA that can be packaged, you'll notice an inversion of B prime, or sorry, of B and C. And if you write out your complementary sequences by hand and duplicate them, you'll see why this is. And so now these progeny can move off to be packaged, and sometimes you'll find this rep protein being packaged with the rest of the virus in the virion, the physical virion itself. And so it might be slightly misleading to say that this rep protein is non-structural. It doesn't make up the structure of the virus, but on occasion you'll find it packaged with the virus. Yeah?
Is this a viral DNA polymerase or a cellular one?
So the question was, are you referring to this?
Yeah.
So the question was, is this a viral polymerase or a cellular DNA polymerase? So the answer to that is actually it's neither, because it's technically not a DNA polymerase. So the DNA polymerase is a cellular DNA polymerase that extends from here. All this rep protein does, I don't want to say all, because it makes it sound like it doesn't do much. what this rep protein does is it introduces single-stranded nicks here, and it can unwind your hairpins. So it mediates a necessary part of, or a necessary step in DNA replication, but it doesn't have DNA polymerase activity. All that activity comes from cellular enzymes.
And the protein, the rep protein itself is viral.
Yeah, so the rep protein itself is viral, and you'll sometimes see it referred to as NS1 for some of these small single-stranded DNA viruses, rep 78, 68, or just rep. Yeah?
Are the hairpin structures sequence-specific for each end?
Are the hairpin structures sequence-specific for each end? So are you asking, is this sequence distinct from this sequence?
No, if they're the same or similar so that the rep protein knows where to go to its hairpins and is able to nick and or perform release activities so that it knows where they are.
So the question is, are these the same sequence? So I believe that they're the same because you have A's and A's here, but these, so the regions of complementarity are the same, A and A, but the hairpins are inverted.
Like inverted.
Right, so again, remember, this is a viral rep protein, but it doesn't technically have any DNA polymerase activity. All that activity comes from what's provided by the cell, which is why being an S phase is so important for these viruses. So this is just a closer look. We kind of already talked about this in answering the last question. What does this rep protein do? Well, it can bind to the origin, which is this location here. It can also bind to this location here. It can also act as a transcriptional activator or repressor, and we'll see that for a different virus here in a little while. It also has helicase activity, so it can unwind those ends. It's a specific endonuclease, so it introduces a cut at a specific site. Remember, that's a single-stranded nick. It's not a double-stranded break. It can serve as a transcriptional activator. It can arrest these cells in the S phase so that these viruses are able to be replicated efficiently. And again, like I mentioned, sometimes you'll find these being packaged into the virion. All right, so I mentioned that some of these small single-stranded DNA viruses are known as autonomous viruses, and some of them are known as dependoviruses. And the dependoviruses are dependent on what's called a helper virus. So the helper virus essentially will push cells either into or back into the S phase so that these dependoviruses can replicate as well. So the job of helper viruses as far as a dependovirus is concerned is simply that they activate the cell cycle. So here's, again, from molecular biology, our friends RB, retinoblastoma, E2F, HDAC. So these, when they're all bound, they repress S-phase genes. When RB becomes phosphorylated by CDK, cyclin-dependent kinase, it dissociates, and then you get transcription of S-phase genes. So that's a normal, healthy cell cycle process. These helper viruses have proteins that can sequester RB and steal it away from this complex, which will also activate the cell cycle, push these cells into S phase where the dependovirus and the helper virus will both then be able to replicate. Remember, the autonomous viruses, the autonomous parvoviruses, those also aren't capable of pushing cells into the S phase, but they simply hang out and wait for the cell to enter S phase. There's a couple other things that these helper viruses provide. Probably the one that people are most familiar with is the blocking of PKR, which is a antiviral defense mechanism found in cells. So an example of a dependovirus, again, is the adeno-associated, yeah.
Why don't the dependoviruses just sit around and wait for S phase, like the autonomous ones?
That's a good question, and I do not know. Do you know, Dr. Stedman? There you go.
That's what you're getting on the test.
So when these dependoviruses don't have a helper virus co-infecting, they can integrate into the genome. So you get typical replication of that first strand of the genome, and then somehow, and it's unclear how, these viruses can integrate into genomes. Perhaps it's mediated by that rep protein that we looked at earlier, the NS1 protein, or maybe cellular endonucleases. So because these small single-stranded DNA viruses are oncotrophic, right? They're not oncogenic, but they do really need and really like cells to be in the S phase. You can imagine that they may be valuable as treatment for rapidly dividing cells, also known as cancer. So parvovirus infection doesn't induce interferon response in cancer cells, and so this infection in cancer cells could go largely uncontested by the cancer cells. The NS1 protein that we looked at in terms of transcriptional activator is toxic to cells. These terminal hairpins that small single-stranded DNA viruses have can lead to the DNA damage response, which eventually leads to programmed cell death, and they may have some gene therapy applications. Okay, so that's it for small single-stranded DNA viruses of mammals and humans. Yeah?
If we go this so the small one that is able to hang out,
Mm-hmm.
it goes through both lytic and lysogenic phase.
So the question was for the dependoviruses, can they go through a lytic and lysogenic?
But the lytic version would actually be when it's coupled in the right circumstance. Like the lytic would be
Right.
when this goes through.
Right. So in order for these cells to enter a lytic cycle, the helper virus would have to be present. Does that answer your question?
I'm mostly curious because lambda actually has a larger genome.
Mm-hmm.
It has like the proteins that is necessary to not only integrate itself into
Mm-hmm.
the host genome, but also to be able to, come out.
Right.
with the excision. So how does it? So the single-stranded is 5 kilobases, right?
Mm-hmm.
So that's a really that's small.
Right.
So what does it have? Does it have all the proteins that are necessary to do that, or how?
So the question is, do these small genomes have the necessary proteins to jump in and out? And as far as I know, if we back up here, how these viruses jump in and out isn't clear.
Oh.
How they integrate or jump back out of a genome isn't entirely clear. Okay, so we've seen the important role that hairpin ends play in DNA replication, how those solve the problem of how to replicate DNA ends. We briefly talked about susceptible versus permissive cells, which I butchered a little bit, but the presentation will be up online. We really talked a lot about how these viruses are dependent on cells being in the S phase, again, because that's when you have DNA replication enzymes present, and we saw that these viruses can potentially, through mechanisms that aren't fully understood as far as I know, can sometimes integrate. Okay, so we're going to change gears now and talk about more single-stranded DNA viruses, but different single-stranded DNA viruses. So first we're going to talk about some well-characterized plant single-stranded DNA viruses, and then we're going to talk about some really interesting, strange single-stranded DNA viruses that we work on in our lab. All right, so single-stranded DNA viruses of plants are relatively rare. Most viruses that infect plants actually have RNA genomes. So the the single-stranded DNA viruses of plants that we're going to look at are called Geminiviruses, and why they're called Gemini will become apparent here in a minute. So they induce and cause relatively economically important plant diseases, mostly right now in the developing world. So they cause these yellowing leaf curls. So plant viruses are really nice because the way that they seem to be named is you just come up with what they do to a plant and then put the name virus in back of it. And so this symptom here is caused by the tomato yellow leaf curl virus, because you have tomato leaves that become yellow and curl. This disease is caused by the red blotch virus. So these are grapevines that have red blotches. And this is, again, those leaf curl symptoms showing up on a pepper plant. And so this leads to losses for farmers, reduced yields, and crop death. So these Geminiviruses are probably one of the oldest or earliest observed viruses. So there's reports, and some people have speculated that a Japanese poem written a few thousand years ago was actually describing the result of a viral infection. So this Japanese poet described autumn colors in a plant seen during summer. And so these are typical of Geminivirus infections because you get yellowing of plants, which is associated with fall senescence. But it wasn't until nearly 2,000 years later that the nature of these virions in the genome was actually elucidated. So why are these viruses called Geminiviruses? It's because they have twinned, see 1, 2, 2 T equals 1 incomplete icosahedra joined at a quote-unquote missing capsid protein subunit. And so here you have the structure, single-stranded DNA genome. We'll see that this isn't always 100% true here. And you have an electron micrograph that shows your twinned virion particles. So I think that these have really interesting, really cool morphology, which is part of the reason that they're in today's lecture. The other part will become clearer later, hopefully. So these genomes, so on the previous slide, genomes represented as one circular piece of single-stranded DNA. But in reality, some of these Geminivirus genomes can actually have bipartite genomes. So you can have DNA A and DNA B. On DNA A, you have the rep protein, or AC1, which mediates replication similar to the rep protein we already discussed by nicking and joining DNA. No DNA polymerase activity. This replication is by a mechanism called rolling circle replication, and we'll come back to that in a couple slides and look at it in a little more detail. This rep protein can also, just like some of the other viruses we looked at, can also push cells back into the S phase by kind of that same mechanism we looked at earlier by sequestering the RB protein. AC2 and AC4 suppress gene silencing. So gene silencing is one of the mechanisms that plants use to fend off, fight off viral infection. Also on DNA A is the capsid protein. The capsid protein for Geminiviruses determines which insects are able to move these viruses from one plant to another. On DNA B, you have open reading frames and proteins involved in movement within one given plant. So because plant cells are connected by junctions called plasmodesmata, viruses within a plant can move without the need for this capsid protein. So these proteins are encoded by DNA B. One of them is known as the movement protein. And so these movement proteins are able to shuttle this genome around to new cells where they initiate an infection without the technical need for a capsid protein. Again, this capsid protein is important for getting this virus out of one plant and back into a new host. so this final note here is that this rep protein can initiate DNA replication of both of these DNAs, both DNA A and DNA B, in this constant region. All right, so those are small single-stranded DNA viruses of plants. Now, the last small single-stranded DNA viruses of animals that we'll look at is Porcine Circovirus. So again, very small, circular, single-stranded DNA virus. Circoviruses generally have two open reading frames. One for the capsid, forms the structure of the virion, and one for the rep protein, which mediates DNA replication. And these porcine circoviruses cause economically important diseases in pigs, so mostly a post-weaning type disease. All right, so we'll briefly look at rolling circle replication now. So this is applicable to the circoviruses and the Geminiviruses. So for circovirus replication, one protein is totally required, and that's the rep protein. You actually find two rep transcripts that are found when you look at infected porcine cells in vivo. But both of these rep transcripts seem to have the same function, and that function is simply DNA nicking at an origin. Cellular polymerases come along, replicate, and then this rep protein joins or completes DNA replication of the viral genome. So that's one domain of the protein. The other domain is a helicase domain. This is likely necessary for binding DNA and then unwinding a supercoiled form of DNA. And so this is just to show these in vitro assays. What people have done is they've taken purified recombinant rep protein, and they've incubated it with DNA that has the correct sequence, the correct structure of the origin of replication for these circoviruses. And what you'll notice is that this larger piece of the origin DNA here is cut or nicked by this rep protein, resulting in the appearance of a smaller band on this gel. And you'll see that both rep and the rep prime, which is the other transcript, the other protein that's found in cells, they both seem to have the same function, which is nicking DNA. All right, so graphical representation of rolling circle replication, which is our second distinct single-stranded DNA replication method today. So here's our infecting single-stranded DNA virus. Here is a stem loop, which is the origin of replication. Somehow, in cells, this single-stranded DNA is converted to double-stranded DNA. So now this form here can serve as a template for transcription, or we can make more genomes. So at this sequence and structure in the stem loop, the origin, that rep protein is going to introduce a single-stranded nick similar to what we saw for our earlier single-stranded DNA viruses. It's going to expose a free 3' OH. Then cellular polymerases can come along and make a new genome. And this rep protein, again, just like with our other examples, it stays associated with the 5' end as a new genome is made here in blue. So here's our new genome being made. We're copying. When we get to the end here, the rep protein can mediate the rejoining of this 5' and 3' ends of the green genome, which can then be released, and this whole process can start again. All right, so Geminiviruses and circoviruses are neat because of, for the Geminiviruses, the diseases that they cause, they're twin dicosahedral capsids. Circoviruses are interesting because of rolling circle replication, but on their face, I think that they're less interesting than these next viruses that we'll talk about. So this is a picture of Boiling Springs Lake here, which is a lake in Lassen Volcanic National Park, which is in California, 50 or so miles east of Redding. So it might be generous to call this a lake in the traditional sense because this is actually a really hot, 50 degrees Celsius, really acidic lake. So for some of the stuff that Dr. Stedman's probably told you about our work, this is relatively almost cold. So a lot of our viruses come from 80 degrees Celsius boiling acid mud pits. So this is relatively tame as far as the places that our viruses come from. But this lake, this hot acidic pond maybe, only contains life from microbial sources from all three domains. When you have microbial life, you also likely have viruses. And so one of the questions that Dr. Stedman and his former students asked were, what kind of viruses are there actually in Boiling Springs Lake? So what they did was what's called a metagenomic study, in this case a metaviromic study. And so a metagenomic study refers to going into an environment, extracting the DNA in that environment, sequencing the DNA in that environment, and then being able to answer questions about what types of microorganisms are here, what are those microorganisms doing. When we look at a metavirome, we select only for viral nucleic acids, and we can do that just by size excluding cellular life. So one of Dr. Stedman's former students, Jeff Deemer, sequenced all the DNA, all the viral DNA in Boiling Springs Lake, and then he assembled viral genomes, and he came up with this graphical representation of what's going on in this lake for our DNA viruses. So we have a lot of double-stranded DNA viruses that infect archaea, bacteria, eukarya. Yeah, that's all expected. We have some single-stranded circular viruses that infect bacteria, archaea, eukarya. That's all expected. But in this little sliver here, he found sequences that looked like they came from a single-stranded RNA source. So why would you be finding RNA-like sequences in samples that should only contain DNA? And so as Dr. Stedman tells the story, Jeff brought this to him and said, this is really interesting. What do you think is going on here? And Dr. Stedman said, well, why don't you go back and check, make sure that that's actually single-stranded DNA. And so he went back and checked. Turns out, yes, it's actually single-stranded DNA. What eventually they were able to pull out of Boiling Springs Lake was this genome that's now been called Boiling Springs Lake RNA-DNA hybrid virus. And so the reason that Jeff was seeing RNA-like sequences in a DNA metavirome was that this virus, who has a single-stranded DNA genome, 4.1 kb, its capsid protein is homologous to capsid proteins found in Tombus viruses, which are single-stranded RNA viruses that infect plants. So a capsid protein homologous to single-stranded RNA viruses, but the other open reading frame, or sorry, one of the other open reading frames in this virus is a rep protein that seems to be from, or it's homologous to those rep proteins found in circoviruses, which we know are single-stranded DNA viruses. There's two other open reading frames in the genome that don't have homology to anything in the databases. And then conveniently, there's also a porcine circovirus-like stem loop here, which also serves as the likely origin of replication for this virus, which makes sense because we have a circovirus-like rep protein. So you can see similarities in structure and sequence between the stem loop from porcine circovirus and from Boiling Springs Lake RDHV. So Dr. Stedman and Jeff, I guess I should say Dr. Deemer, published this paper in 2012. And again, as Dr. Stedman says, they sent it to different places, and some of the places sent back the paper and said either no, we don't believe this, or rejected it. So they sent it eventually to Biology Direct, and they won a bunch of awards through Biology Direct, Paper of the Year, and so on. And then at different meetings, people have come up to Dr. Stedman and asked him, why didn't you send that paper to us? And he says, well, we did, but you didn't want it. So this was the first published, recognized example of seemingly recombination between RNA and DNA viruses. So how might this have happened? So here's our BSL-RDHV genome. Here's our rep protein. Here's an initial single-stranded DNA-like capsid protein. And somehow we think that, and Dr. Nacho, who gave you another lecture, has other ideas maybe he'd explain to you or talk with you about, but somehow this single-stranded DNA virus likely obtained a single-stranded RNA capsid protein. Probably physical RNA was incorporated into this genome somehow. So now you have a genome that's truly likely part DNA here, part RNA here. And by reverse transcription, this RNA is converted to DNA. And so now you have a DNA-like capsid protein and an RNA-like capsid protein present in the same virus. This DNA capsid protein is then lost by a deletion event, and you're left with a single-stranded DNA virus that has an RNA-like capsid protein. So Dr. Stedman's proposed a couple different mechanisms by which this event might have happened, and what I'm interested in is potentially how the rep protein here may have mediated this capture. Because we're running a little short, I think I'll go quickly through this. And so Dr. Stedman laid out these different mechanisms in one of his papers that you can find here. So I mentioned that this was the first published definitive example of likely recombination between RNA and DNA viruses. There was a paper that came out, I think, a few months, maybe a year before this paper, the original RDHV paper, in which a group from Korea studied the viromes of Korean air. And they found RNA sequences in their DNA metaviromes. What they did was they essentially threw those out and they ignored them. If you look at their figures, in that paper they have them largely pushed to the side, and they largely ignore those. But since then, people have gone back and started looking through old metaviromic data, old metagenomic data. People have started looking for these in new studies, and what people have found as of May 2018 is that these are actually, common may not be the right word, but there's a whole lot of these out there in the environment. So I think we're up to around 400 of these sequences. And so what all of these sequences have in common is that they all have a capsid protein from single-stranded RNA viruses, the Tombus viruses, which are plant infecting. So the capsid protein forms a monophyletic group. The rep proteins, though, come from various single-stranded DNA viruses. So the Circoviruses, Geminiviruses, nanoviruses, which are also plant-infecting viruses. You'll find rep proteins from many different single-stranded DNA sources in these genomes. And so what this suggests is that you likely had one instance, if we're operating under the assumption that somehow a DNA virus acquired an RNA sequence, you likely had one instance of recombination between an RNA and DNA virus, one capsid protein acquisition event, followed by subsequent rearrangement of these rep proteins. So what do we do in our lab? Well, another thing that we do in addition to being interested in the rep, is we look for these cruciviruses in different environments. And Ellis and Nacho, who are sitting back there, are responsible for finding 2 new cruciviruses. So I'm calling them something now that I'm realizing I didn't tell you why we're calling them cruciviruses. So the initial name was RNA-DNA hybrid virus, but that has a little bit of a connotation with it that maybe your genome actually is a hybrid of DNA and RNA, which isn't true. These viruses are completely, we assume, single-stranded DNA. And so another group came up with the idea to call these viruses cruciviruses because they represent a cross between RNA and DNA viruses. And so now the accepted name for these is not hybrid viruses. I still refer to our viruses as RDHV. Now the accepted name is technically cruciviruses. So Ellis and Nacho have found 2 of these genomes from the Woodburn peat bog. So there's other studies out of France that for whatever reason show a high abundance of these genomes being found in peat bog and fen environments. And Ellis and Nacho will tell you that the Woodburn peat bog is actually more of a ditch now. It was a historical peat bog, but now it's more of a canal. But they were able to find 2 of these crucivirus genomes there. Late last year, so initially when these viruses were described and have been described, it's just the genomes that we've been finding and sequencing. And so we don't know what these viruses are infecting, but late last year there was a paper that came out that found that these cruciviruses seem to be associated with marine isopods, and luckily for us, these marine isopods that this group was looking at, and this group happened to be from the East Coast, they were looking at marine isopods in the Pacific Northwest. And so they looked at isopods on the Canadian coast, off the Washington coast, and I think off the California coast, which I guess isn't Pacific Northwest. At any rate, they found these crucivirus genomes associated with these isopods. And the takeaway here is that when they did qPCR to look at how often these genomes show up in specific spots, their conclusion was that these genomes are likely associated with unicellular protozoans that in turn associate with the isopods. All right, so what else are we interested in our lab related to these cruciviruses? One of the big questions is, how does a single-stranded RNA capsid package a single-stranded DNA genome? So that's one of the things that Dr. Nacho, who gave a lecture earlier, is trying to answer. We're interested in the biogeography of these viruses. Where do they show up? Why are they there? What types of evolutionary relationships can we infer from the sequences that we do have? Mechanisms of recombination or other hypotheses about how these viruses arose. Structures, what do these viruses look like when they're assembled? What does the actual virion look like? What's the host? What do these other open reading frames do? What does that rep protein do? And the list kind of goes on and on, potentially. Okay, so that's the last slide about viruses, but there's one kind of overarching question in an evolutionary sense of why would something have a single-stranded DNA genome? Because it seems, at least on its face, that existing in a single-stranded DNA form would be inefficient, right? Because single-stranded DNA is a target for degradation. You elicit all kinds of emergency responses in cells, right, when you have single-stranded DNA present. So it would seem that this would be a relatively inefficient way to exist. Perhaps the cruciviruses offer one clue as to why this might be an efficient way to exist. Perhaps single-stranded DNA viruses possess a unique mechanism by which they can recombine with other viruses rapidly and quickly, particularly potentially RNA viruses. And by doing so, they're able to sample the landscape of stuff that's out there. Right? So if you can rearrange and obtain a new capsid, you can likely influence your host range. And if you can find a new host that you haven't been evolving with for millions of years, that's likely an evolutionary advantage because that host won't be ready to counter your genome or your approaches to replicating your genome. So potentially, maybe these single-stranded DNA viruses are able to quickly access new and broader host ranges, which allows them to avoid immune responses or detection by cells. All right, so I guess now this should probably be changed to Cruciviruses because that's the accepted name, but I still say RDHV. So we talked about the Boiling Springs Lake RNA DNA hybrid virus. We talked about potential origins. We talked about some of the stuff that we do in our lab. We looked at rolling circle replication in the circoviruses, and that's also applicable to the Geminiviruses. We briefly talked about RNA capture and why existing as a single-stranded DNA virus might be evolutionarily advantageous. So that's all that I have for you guys. So if you guys have questions, I'll do my best to answer them and not confuse you.